सॉकेट एक ही है, दो सॉकेट मोबाइल वाला देखो ना, वो चलो लैपटॉप यहाँ रख दो। आते ही समय से पहले मर्दों को टेस्टिंग इसको ही देना
उनका डेटा भी फुल हो गया था इसकी वजह से।
ये तो फिर हमारा चैनल पे है तो फिर आराम से हो ही जाएगा।
हाँ वो तो हो जाएगा।
हाँ बोलिए।
जस्ट अ मिनट, जो स्लाइड ऑन करते ही चलो। हेलो, हाँ हाँ कोई बात नहीं। मैं इधर चालू करके उधर ठीक है। या
Good afternoon to one and all. Welcome back. Moving on to the next session, we have Dr. Elizabeth Lipke, Professor, Department of Chemistry, Chemical Engineering, Auburn University, USA, to share some insights on the topic Engineered Tissues for Regenerative Medicine and Disease Modeling Applications. And we have Dr. Anand Deshmukh, Professor, Academic Head and Department Head, Regulatory Affairs, STPC KIM, to chair the session.
No, this mic is not required. You can even follow that. You can tell her what is the slide change. बोलो। बोलो, क्या है?
Which one?
You tell her.
Good afternoon, everyone. It gives me an immense pleasure to welcome you all back to this session of our 7th International Conference. This session promises to be both insightful and inspiring, focusing on the exciting field of engineered tissues for regenerative medicine and disease modeling application. We are privileged to have with us Dr. Elizabeth Lipke, the George E. and Doughty Stafford Olath Professor from the Department of Chemical Engineering at Auburn University, Alabama, USA. Dr. Lipke is an internationally renowned researcher whose groundbreaking research is the intersection of biomimetic materials, stem cell biology, and tissue engineering. Dr. Lipke's career is a testament of her relentless pursuit of innovation. She earned her PhD at Rice University, followed by a prestigious postdoctoral fellowship at Johns Hopkins School of Medicine. Her research has significantly advanced our understanding of how biomimetic materials can guide pluripotent stem cell differentiation and how these principles can be applied to creation of cardiac and cancer tissues. Her research group has developed techniques to encapsulate high cell densities within microspheres, enabling the formation of dense functional engineered tissues. This pioneering work has applications in cardiac tissue engineering, therapeutic cell delivery, and cancer modeling. Dr. Lipke's contributions have been recognized globally with numerous awards, to name few, a National Science Foundation Career Award, and her induction into American Institute of Medical and Biological Engineering College of Fellows. Without further ado, it's my distinct honor to welcome Dr. Elizabeth A. Lipke to deliver her talk entitled Engineered Tissue for Regenerative Medicine and Disease Modeling Application. Please join me in giving her a warm welcome.
Thank you very much for having me here, for the invitation to come and talk to you all. It's been a very exciting day and really exciting opportunity to see your campus and to see everything that's going on here. I'm going to talk to you today about something that's a little different than what perhaps what you have been studying or what you see typically, but that's the formation of engineered microtissues. So as we all probably probably realize cardiac regeneration is not possible currently today with drug treatments. We have many major diseases that contribute to heart disease, but in the end, we cannot regenerate the heart. And when we start to think about cancer, the cancer microenvironment influences the way that cancer cells respond to drugs. And so we need to be able to better mimic that in order to come up with and better understand the mechanisms of disease, as well as identify better therapeutics. And so my research group at Auburn University works on these two areas, and basically interfacing between the in vitro and the in vivo. So models that grow in a dish, and how we can use those models to grow in a dish to inform further steps. So there's some questions that you might be asking. Why engineer a tissue? And I guess we're not talking about those things that you use for your nose, right? We're talking about actual human tissues. And so, sorry, a little bit distracted. And so when we start to think about this, I chose this picture because actually one of my students, former students, Shantanu Patman, is in the back looking at his cancer tissues, and another one of my students, Alex Hodge, is in front looking at his cardiac tissues. And so there's two reasons people make engineered tissues. One is for regenerative medicine, and the other is for drug discovery and development, and so modeling disease in a dish. And so in order to do this, we take materials and we bring them together with cells and bioactive factors. So when you think about a cell in your body, it's receiving all different types of cues from your body about how it should behave, what it should do, what it has to eat, how it should react. So it's kind of like you when you lived at your parents' house, right? There was all types of rules, things you had to do, things you were going to eat, the way you got your food, the way you got to places you wanted to go. And then, maybe you moved to live in a dorm, and some of your behavior might have changed. And this is the same thing as a cell. When the cell is living in our body, it's getting all types of cues from your body. How it should behave, how it's going to live, what it's going to do. But when we take that cell out of the body and put it into a dish in order to study it, it's no longer getting those messages. It doesn't have the same environment around it, the same extracellular matrix, the same stiffness, the same neighbors, right? If you move, your friends change, and that might change what you're interested in or what kind of food you eat. Same thing for the cell. When the cell is surrounded by different neighbors, it may decide to behave differently. And so in order to better model disease and understand disease, we want to add those neighbors back in. We want to add that extracellular matrix or stiffness of the environment around the cell back into the equation to see if we can create models in a dish that are more similar to the environment, the microenvironment that the cell had inside the body. Just like your parents, when you moved out of their house, tried to get you to behave the way you did at home, right? We're trying to get the cells to behave the way that they did in the body so that we can understand those processes that happen inside the body, but do it outside in a dish. And so this is the concept of tissue engineering and how we're working to build these models. And so we do that with these materials that I'm going to tell you a little bit about, the bioactive materials. We have cells, different cell types or combinations of cell types, and then we have bioactive factors. And so those bioactive factors could be, think about these like text messages, right? They could be cytokines, they could be, in our case, the microenvironment from the diabetic microenvironment, insulin-resistant microenvironment. What is it that the cell is exposed to, and what we'd like to understand how the cell behaves in that environment. And so then we can add even pathological inputs, which would be the diabetic microenvironment. And we use these things for things like drug testing, understanding human development, mechanisms of disease, and eventually for identifying therapeutic interventions. Materials matter, and so in our lab we're really interested particularly in how the materials might influence the cell's behavior. When you think about materials or extracellular matrix, we have binding sites for cells. So that's like the floor, right? You walk on the floor. If you had to be just walking on your hands and knees, you'd behave differently. But also degradation sites, so the cells can remodel the material and make their own extracellular matrix. How stretchy the material is influences behavior. Even the nanotopography influences how the cells behave in their environment. It's kind of like the doors and windows in the room. If you lived in here without doors and windows, you behave differently than if you can change the environment around you, if you can open the door to go to a different place, or if you all of a sudden live on the 3rd floor of a dorm as opposed to living on the flat or not having an elevator, right? You're going to change in your behavior. Same thing for the cell. And so even electrically conductive materials are some of the things that we've looked at in my research group. So the advantages of using biomimetic materials to form these tissues are several. One is that we can provide these physiological cues, like the electrical conductivity to the cells. Another is that we can create an environment where every cell has as similar environment as possible, especially when we're considering how to do drug testing. Each tissue that we make has to be the same as every other tissue, so that that individual tissue in that individual well is the same every single time we make it, so that when somebody goes to test the drug, the response that they're getting is the drug that they're testing, and not because we had variability in our tissue or the cell was behaving differently in that space. So by providing the cell with the material, the cell has a uniform environment that it's living in, it's more likely to do the same thing. Another way is to support the cell in a scalable system. So when we want to make enough cells for a particular therapeutic intervention, for example, we need to scale up production. We can't just create the cells in a little flask, we have to grow them in a big bioreactor, and we want to be able to do this in what we call suspension systems. So systems that are stirred flask. And then our lab is also interested in implantation of cells, and so having a material provides the cell with basically an encapsulated system when you're injecting it, so that it's not exposed to the shear stress, to the mechanical forces that happen when a cell goes through a needle that cause it to die. In particular, the studies I'm going to show you today use a photo-cross-linkable hydrogel system. This is a hybrid biomaterial, so it has a synthetic component and a natural component. The synthetic component allows us to manipulate the stiffness and allows us to have it go from a liquid to a solid very, very rapidly. In our system, we can do that in as little as 1 second. The natural component, fibrinogen, can be degraded, and it provides cell adhesion sites for the cells so they can hang on, and also they can remove it and replace it with their own cell-secreted matrix. So why spheroids? Why did we do, like many people do tissue engineering, we're very interested in microspheroids. This is one of the special things that we do in our lab that other people don't do. And it's for 3 specific reasons. One is cell production. As I mentioned, we would like to be able to scale up the production of these cells into bioreactor systems to make enough cells. The second is cell delivery, so to provide that injectable format. And the third is drug testing and disease modeling, which is what I'm going to be focused on today. And so this makes these microspheroid systems are similar to the systems that Dr. Arnold spoke about earlier where he had cells, but they're suitable for high throughput drug screening. And so these are just some of the papers that we've published in each of these areas. So as you know, there are some challenges in drug development, and this includes identification of new candidate models, and specifically in cancer models, as you go from 2D cell culture to animals, and then animals to humans, there's a disconnect. And so the in vitro model systems are looking to bridge this gap and provide information that 2D cell cultures can't provide and animals can't provide. So basically giving additional information that can be used in the drug discovery and development process. When we think about 2D cell cultures versus 3D cell cultures, the microenvironment the cells are in matters. So when cells are grown as a flat monolayer, they have a limited amount of space that they can grow in. When you put them into a 3D culture, you can grow them for longer periods of time, so they're not limited by the 2D space available. The other thing is that the cells in 2D culture are very easy to analyze, so we have to keep that in mind. As we move to a 3D system, it's much more complex, and so the imaging and the analysis is more difficult. But the 2D cell culture does not represent the microenvironment, like I mentioned. So in the cell microenvironment, we have multiple cell types. We have cancer cells, We have fibroblasts, we have immune cells, a range of immune cells. We have vasculature and extracellular matrix. And so the 2D cell culture environment does not incorporate those things. Whereas the 3D cell culture, we can manipulate to incorporate those pieces. So in cancer cell tissue engineering, we've done a lot of different types of studies to mimic or recapitulate different aspects of these systems. And so we started out making tumor millibeads, which are these spheroids here. So they're large enough so you have a hypoxic core. You can see them here. We looked at how matrix influences cells. We've looked at the microfluidic systems that Dr. Arnold mentioned. Today I'm going to focus on these tumor microspheroids. And currently we're working on now going from patient-derived cells and incorporating and making these tumor microspheroids with patient cells and patient-derived xenograft cells. I won't be covering that, but I'm happy to answer any questions you have about that process later. And so I specifically actually focused on this because this work was done by my former graduate student, now faculty member at IIT Madras, Shantanu Pradhan. And so what he did is developed a novel technique for making these 3D cultured microspheroids and compared them to self-aggregated spheroids. And so in this case, he used, at the top, pegfibrinogen material and photoprosmic dip. In this case, we were using a vortex system to produce these microspheroids, and we compared it to aggregates. And so these are MCF7 cells, which are a self-aggregating cell type. Not all cancer cell types aggregate. And so this is a big challenge, is if you can't get the cancer cell types to aggregate, then you can't do it this way. So we chose an aggregating cell line. And what we found is when you look at, in red is each green and blue, each batch, and then each dot represents the cell density in a different spheroid. And what you can see is, in the tumor spheroids, the way we produce them, even at day 3, there is variability within the batch and somewhat variability between batches. And as time progresses, we have ever higher variability in the size and the number of cells per volume of these two aggregates. And this is also between batches and within batches. And when you make these with the material, you see that you have very tight control, and this was a vortexing method, so not very effective in terms of typically getting the same size even. You have tight control over the number of cells in each spheroid, and this is very reproducible between batches, both at the beginning, and it continues to be reproducible over 21 days of cell culture. And so here's just a look at the ultrastructure of what these cells look like when they aggregate versus when the material is incorporated with them. So when you look at the top, you see these very smooth tumor spheroids. All the cells are basically adhering to each other. When you incorporate the material, similarly to the ECM, the cells are disorganized and don't have that smooth appearance. And so here, when we started to look at the fluorescent staining, you can see that here is the aggregated cell, and you see the MCF7 cells have this pattern morphology, more similar to normal breast epithelium. And when you incorporate the material, they become disorganized, much more similar in morphology to the carcinoma. And Dr. Pradhan analyzed this looking at apical basal polarity as well as nuclear area, and showed that there were differences between these spheroids and the tumor microspheres. Now you can see from the picture, even in these microspheres, the way we were making them here with vortexing, there's still variability in the size of these spheroids with the cancer cells incorporated. And so what another graduate student and then postdoc in my research group, Aaron Cito, asked is, can we rapidly produce these in a highly uniform way? Can we make them very quickly and very, very similarly? Long story short, as you can see from the picture, yes, he could. Right? These are all his spheroids. He made them. And they're all the same size and all the same shape. So how did he do that? So basically, we built this built this microfluidic device that is able to produce these cell-laden microspheres. The polymer precursor that I told you about comes in from the top. In this case, mixed with many different cell types that we've tested. It goes down into this chamber. The mineral oil comes in from the bottom, and we photocross link using visible light here. And then we flow the cell culture media and collect our spheroids at the end. And so you can see as the polymer is exposed to light, it goes from being a liquid, like I mentioned before, to being a solid. And that happens in 1 second. And so this is just what it looks like. Oops. I'm hoping. Is there a way to play the video or a way for you to click the video? Sorry. Can you click the video? Thank you. So this is what it looks like in real life. These little spheroids progress down that little channel that we form, and then they go from being a liquid to being a solid. So we can make 60 microspheres in a minute, and we can vary the diameter and the cellularity. Each of these has, if you have 800 micron diameter, 5,000 cells each, but we can have up to many more cells each, depending on the cell density. This is what it looks like in the lab, all set up. And basically what that means is in less than 2 minutes, we can encapsulate 1 million cells. So it's very important both for if you want to put cells into a bioreactor to produce a lot of cells, and if you want to just make a lot of cells very quickly for doing an assay. So these are very high cell densities. For reference, most people put about 1 million cells per mil into their engineered tissues. We're doing 10 million to 60 million cells per mil. So we change that cell density depending on the application and how long we want the cells to grow and what types of cells we want. We can also mix 2 different cell types together. They don't have to like each other. They don't have to want to be together because we're photocrossing the material. They can stay together the way we put them. So you can see here we have very high consistent diameter, circularity, both within batches and between batches, which is really important for the long-term applications. We have really good cell density. We've encapsulated many different types of cells. These are equine endothelial colony-forming cells that we use for in vivo large animal studies for wound healing. The MCF7s, MD-MB231s, 2 different types of breast cancer cell lines. These are HIPSCs. We've done patient-derived xenograft colorectal cancer cells, as well as others. So for example, for our breast cancer tissue microspheres, here you can see they're growing from day 0 at the top to day 14 at the bottom. Again, they have good reproducibility in terms of the microspheres themselves. They are remodeling their extracellular matrix. They become stiffer. CMCF7 cells do. The MD-MB231s are more metastatic cell type, and so they're actually more migratory in nature. And what we did with these then is sent them off, so we can load these into 384 well plates or into 96 well plates using an automated system. And then we ship them off, and people use them for drug testing. So they add the drugs using an automated system into the well plates, monitor them over time, and this is just some pictures of them in the well plates. And we've also published, and you can look at the publication of how we do that and how we can look at the cells. So I'd like to also tell you a little bit about our cardiac work. So one of these questions we asked is, can we also encapsulate pluripotent stem cells into these microspheroids and make them into heart cells, or cardiomyocytes? And so this work was started by Petra Kirscher on the left, who's now at Merck in Darmstadt in Germany, and finished by Bertus Finkley on the right, who is working for Pharmavite in their process development. Basically, she makes gummies and learns, and she helps develop processes for making gummy vitamins. And so what they did is they took HIPSCs, or human-induced pluripotent stem cells, which are genetically reprogrammed cells. So if you think about it, it's like taking cells that are PhDs or PharmDs, sending them all the way back to kindergarten genetically, or first grade, and then training them again. And so we have to train them. And just like maybe your parents pushed you to be here, and they thought, this is a good career path for you, we need to push all of the cells to become one thing. And so that's what we're trying to see, is can we push the cells after they've been genetically reprogrammed into HFPSCs to become just one thing. And so some of the problems for cardiomyocyte use in cardiac tissue production are the fact that when you take cardiomyocytes contracting heart cells apart, they die. They don't survive this dissociation process. And so we want to be able to scalably, uniformly produce them and get them to mature to the point that they can be used for drug testing and specifically look at things like arrhythmias, which is a big problem when you're considering, but you have to have mature cardiomyocytes to do that. And so we need large numbers of cardiomyocytes with low arrhythmogenic potential. And so some of the work in our lab is looking at many different ways of doing this. I'm going to show you just a little bit about the process that we're using. And so, like I said, we want to go from 2D cell production to 3D cell production. But most times people make these cardiomyocytes, they take them apart, and then they try to put them back together. And they electrically, mechanically stimulate them, they try to tweak them to get them to come back together. It's kind of like taking everybody you know and putting them in a new city and telling them to make new friends. It doesn't work very well. And so what we found is that by taking the stem cells and having them differentiate or go through the developmental process all together, we can make these engineered tissues without having to take them back apart. And this is much more efficient from a production standpoint and also from a maturation standpoint, as we see. So what we do is we mix again these pluripotent stem cells, the ones that got sent back to the first grain, with the material. We photo cross-link them, and then we provide them with small molecules that direct their development from being pluripotent, able to become just about anything in the body, to becoming cardiomyocytes or heart cells. And so we do this using a very standard protocol. And what we find is that in the 3D environment, we have the ability to make these, what we call engineered developing cardiac tissues that develop over time. And so on the left, you see day 15, and on the right, you see 3 months. And so you can see the cells elongate. So they are, you can see that they begin to have connexin 43, which is a gap junction on the apical ends of the cells, and they become more mature over time in these tissues. Much more mature, in fact, than anybody was able at that time to be able to show with taking cells apart and trying to put them back together. And so what we see is that we have aligned sarcomeres with C-bands and mitochondria, showing this, and that we have T-tubule formation. And so, in fact, this was never shown before this with human-induced pluripotent stem cell differentiation, that they were able to form tubules. And it's because we allow the cells to develop together. And so those developing cells, as they develop together, are able to form these important junctions that, no idea what I did. Did I do anything? My fault. Right. That they're able to form these junctions, and because we don't tear those junctions apart and try to get them to reform those junctions, the junctions are able to mature. And so just to look at them here, we did this in a whole bunch of different geometries, but including in the spheroids. And so you can see here the spheroids. Here's the cells forming dense tissues over time. We have high cardiomyocyte content, so we actually have 70% cardiomyocytes. We have some endothelial cells, we've shown that by single-cell RNA-seq, as well as some fibroblasts, so very smooth muscle cells, so very typical composition that has been found by other people is the cell types you have to add together to make good engineered cardiac tissues. These spheroids have dense cardiac tissue throughout them, and they have alignment that is indicative of them becoming a mature cardiac tissue. So on the left, you see this day 60 microsphere. We can keep these in a dish up to several years and just allow them to contract and grow and look at them. My students never tell me how long they've kept them because they think that they tell me that they'll die. But right, it's always the instructor or the faculty member messes things up. And in the middle, you see human cardiac tissue. So they are very similar in terms of some indicative of their structure. We see appropriate changes in gene expression over time. And importantly, they respond to electrical and pharmacological stimuli, so we can pace them, for example, up to 6 hertz, and give them beta agonists and antagonists, and they respond appropriately. So finally, we wanted to be able to control the size and shape of these microtissues, right? So some of you might be thinking, well, right, these are contracting cells. And I think, you know what, I think we should have, sorry. Yeah, can you click on that video also just so that we can see? So I told you a lot of times they move, but it's much more impressive to see them. There's some others, but we won't go back that far. So they contract, right? So they're contracting, like the heart contracts. And so, as you can imagine, when they contract, that's putting different forces on the cells. And so, in the literature, we know that the shape and the size of the tissue influence how the cells align in the tissue. So one of the things we wanted to do is to be able to make different sizes and different shapes of microtissues so we could explore how the functionality of the cells might change in response to shock size and shape. And so, Dr. Tian, Yuan Tian, who's a graduate student in my lab and is now been a member of my lab for a long time as a postdoctoral fellow and researcher, went through and looked at the fluid dynamics in the system. And basically we looked at two different things. I don't know, do I have any chemical engineers? No, nobody's a chemical engineer. Okay. Well, chemical engineers are all about dimensionless groups, right? So we have defined two dimensionless groups. One is the narrowing ratio, which is the ratio of the junction to the ratio of the outlet. And the other is the flow fraction, so the flow rate in the two different inlet streams. And by changing those two numbers, we're able to change both the size of the spherical tissues to make them larger or smaller, and also the axial versus radial diameter to change how long they are versus how wide they are. And so we're able to control all of these aspects. And in order to, by doing that, then we were able to control the cell maturation, which I'm not going to show you because my student is not published that particular thing, you know. But at any rate, so we're able to make these very consistently control the size, and that impacts the cardiac function in the end. So the cells that are in the rods mature much more than the ones that are just in the spheres. And so in conclusion, what I've shown you is that these materials can be used to create 3D engineered tissues that mimic both the tumor microenvironment and support cardiac development. The pluripotent stem cell encapsulation is allowing us to make these 3D engineered cardiac tissues, and we can use these for multiple applications, so cell delivery as well as cell differentiation and drug testing. And so with that, I'd like to thank my group. These are all of many group members, but these are the ones that actually worked on these projects that I showed you today, and many collaborators from many different places, including all of them. I'd be happy to take questions.
Thank you so much, madam, for this wonderful session. Thank you for illuminating us with the thought-provoking presentation about the polymerized PEG fibrinogen hydrogel, if I pronounce it correctly. So here, to bring this story very simple, what the metaphor the madam has used was very fantastic. She said that this bioenvironment are like our neighbors. So, like if we keep our neighbors constant, so our behavior will be very constant. So similarly, by mimicking the microenvironment of the cell, we can regenerate or we can produce a very similar kind of cell. And this kind of technology is very helpful, particularly for the development of a 3D model cell lines. And then this also helps in bridging the gap of an animal and human trial. And to achieve this, she has suggested the use of a microfluidic technology, where there, with a right amount of fluid flow and a right kind of an aperture used, we can reproduce the different size and shape micro space. So within that, the pluripotent stem cells are being incorporated to develop a cardiomyocytes or the cell line. So I could say that where the science meet the part of our hearts, right? So with this, we can say that these are baby hearts which can be definitely been used for the cell delivery, cell differentiation and drug testing purposes. Thank you so much, ma'am, for this wonderful session. Now, I would like to open the floor for the question. Please feel free to share your thoughts and inquiries with Dr. Lee.
Thank you. You have mentioned Yamanaka factors like positive for these things, but semic factor is carcinogenic. If you use that, then that heart is carcinogenic.
Sorry, what is carcinogen?
Semic factor, cofactor. Just like you have used OCT4, SOX2, these things.
Oh, okay. So as the cells differentiate and become cardiomyocytes, they stop expressing those pluripotent genes and start expressing the cardiomyocyte genes. And so it's very important that all of the cells become cardiomyocytes and not express the pluripotency genes. And so currently, I didn't show you, but what we're doing is looking at all of the cells at each time point in the differentiation process. So we started at day 0, day 3, day 5, day 7, day 9, and doing single-cell RNA-seq to identify all of the cell types that are present at each point in differentiation, both to look at how reproducible the cell population is in each batch compared to another batch, and also to understand what cell types are present. And so in all of the batches, when you get, for example, to day 10, we don't have cells anymore that express the pluripotent genes. They're gone. But in 2D cell sheets, for example, when you do these same processes, that's the same thing. But you are very right. You have to be very careful that the process that you develop is not allowing even a single cell to get through that is still a pluripotent stem cell, that is a cardiomyocyte. Now, the other thing is that the cells produce their own matrix as they distinguish, and so they remove the material. I thought that's what you were asking about to begin with, but the material is gone. After 20 days, it's completely gone, so we use mass spec to evaluate the extracellular matrix produced by the cells.
Thank you.
Any other questions?
Very good afternoon, ma'am. As we discussed, I saw that it's a more imaginary scenario when the cell culture grow and come to the final stages of development of the whole organ. Now, as we see that you mentioned that cell grew and attracted the way environment was raised in. Now, imagine the two major questions that I'm leading with is firstly, that in case if the organ is built, how will the body accept it? And will there be rejection from the body in the future if there is a complete organ made? And secondly, as these are cells which are lab grown and it is probably made from stem cells, are the chances of getting genetic mutations in the future?
I think perhaps start with the first thing. Genetic mutations in stem cells, as you culture them, are something one always has to be very careful about. And that requires testing. Your first question, or first part of the question was, how would you have an organ that's available for a person? And so there are some applications currently occurring where they're doing clinical testing, for example, to produce the cells that are forming in the back of your eye, the retinal cells. That's a pretty immune protected space. I don't know that we'll ever get to having whole organ treatments with stem cell generated tissues, just because there's multiple different technologies being developed for replacement of organs, including animal production of organs that can be accepted by humans. But one of the ways we're looking to make larger tissues is by bringing multiple, so we can use these spheroids and pack them together and form macro tissues after we've done the differentiation. and using perfused systems. So we have bioreactor systems that are developed by Synvivo that we can then, or CFDRC is a company, that then perfuse macro tissues to make macro scale tissues to look at things that you couldn't test, for example, in these small tissues, like ischemia reperfusion. You need a larger scale tissue for that. And so those types of things we do by assembling those little spheres into macro tissues by molding and then photocrystalline them together.
Anyone else? Okay, so I can close the session now. Thank you very much.
Thank you so much, Dr. I request you to kindly mark your esteemed presence by signature, like putting a signature on the board.
भाई पीछे आ जाओ भैया.
And I request Dr. Anand Deshmukh to felicitate Dr. Elizabeth Lipke with a memento as a token of gratitude and love. And I request Dr. Uttam More to felicitate Dr. Anand Deshmukh with a memento as a token of gratitude and love for chairing the session. We shall move on to the next session, which is design and development of novel LXR agonist for Alzheimer's disease by Dr. Rajesh Amin, Professor, Department of Drug Discovery and Development, Harrison College of Pharmacy, Urban University, USA. We have Dr. Uttam More, Professor, Department of Pharmaceutical Chemistry, STPC KIM, to chair the session.
Good afternoon to everyone. It is a great honor to introduce our esteemed speaker, Dr. Rajesh Amin, a distinguished professor in the Department of Drug Discovery and Development at Harrison College of Pharmacy, Urban University, USA. Dr. Amin's remarkable academic achievements and groundbreaking research have positioned him as a prominent leader in the field of pharmaceutical sciences. Dr. Amin completed his PhD in pharmaceutical sciences from Wayne State University, Detroit, Michigan in 2003 and undertook advanced postdoctoral training in metabolic therapeutics at Pfizer. Since joining Urban University in 2009, Dr. Amin has played a key role in advancing therapeutic research and development, mentoring numerous researchers and driving impactful projects. His cutting-edge research, supported by the prestigious organizations like the American Heart Association and the National Institute of Health (NIH), has received widespread recognition. His lab is distinguished for integrating medicinal chemistry and pharmacology in biotech-like setup, allowing the development of novel solutions targeting some of the most challenging diseases. Dr. Amin holds patents on innovative compounds targeting nuclear receptors, including liver X receptor (LXRs), which are at the forefront of therapeutic exploration for Alzheimer's disease and frontal temporal dementia. With over $6 million in research funding, his work represents a transformative approach to addressing unmet medical needs in neurodegenerative disorders. Today, Dr. Amin will present his groundbreaking research on design and development of novel LXR agonist for Alzheimer's disease. These talks will explore his innovative compound AU403, which has shown significant promise in delivering therapeutic benefit while mitigating side effects commonly seen with the current treatment. Let us warmly welcome Dr. Rajesh Amin to begin this inspiring session on innovative and discovery. Dr. Amin, the floor is yours.
Keep sure. Okay, that's better. Okay, so a lot of you are graduate students, undergrads, and wondering what you're going to do in life, right? And so, there. So, our ADR came and talked today about Auburn and how Auburn has all these great facilities, are building brand new. The ability to do the research that we do for drug discovery is fantastic there. I love to say we are a football school, but we're not. At least this year, right? We are doing well. It is exciting to go to these games where there are 70, 80,000 people there attending and discussing football. So, my students do the work. My postdoc here does the work. I sit in my office and drink coffee all day. But we work together, as our ADR says, collaborative research, right? So I like to give thanks in the beginning to all the people that have helped attain all this data. And so I talked about my undergrads here, my postdoc, and then my grad students here, and some PharmDs as well. We all work together to help bring in funds and advance our intellectual properties. So today I'm going to talk about a story. It's a little okay, maybe that's a little bit better. Okay, so we're going to talk a little bit about PPARs, and we're going to talk a little bit about nuclear receptors. And that's the area of research which I do. So, that's better. Okay, so I was originally trained in metabolic diseases. I did my PhD in medicinal chemistry and pharmacology. And then I went to Pfizer and I did a postdoc there in the drug discovery department for metabolic diseases. And so we were looking at peroxisome proliferative activating receptors, which are nuclear receptors. They control energy in your body, insulin resistance, diabetes, things like that. And so I became a staple in diabetes research. I then came to Auburn, and I continued this diabetic research, and we designed drugs for PPARs. And I hated our story. So I had a collaborator at Auburn who wanted to do neuroscience. And so I started developing compounds for neuroscience. And this story develops further. So one of the things that I learned is that the incidence of Alzheimer's is just astronomical, right? It's going way out of the limit. And the amount of care and dollars needed in the United States alone exceeds $1 trillion. And so it's an unmet medical need. So we started developing, our compounds for areas associated with dementia. And dementia is a big umbrella where you could have multiple forms of diseases or cognitive impairment. And my lab is focused in on Alzheimer's, as we see here, and frontotemporal dementia. And my postdoc today is going to talk a little bit more about frontotemporal dementia. So one of the things we always have to remember is, when do we give a drug to a patient? Even in animal models, how do we design a drug, and when do we give it? The efficacy of a drug, right? So when we started looking at the drug therapies, we started looking at that you can't give it when a person already has late dementia. We have to diagnose a patient first, then give it. Lilly just came out with a drug or a test for tau and amyloid beta in the cerebrospinal fluid as well as in the blood. And so that's actually now considered a staple for diagnosis of Alzheimer's disease. So previous therapies have all been just associated with not solving the problem, but just delaying the progression. And so those don't work. So we start treatment in this early stage with mild cognitive impairment, as we see here. And then we slowly prevent this progression of the disease for severity. And hopefully the person can last and have a good life. So my lab. Sorry about that. I think my computer is moving this around a little too much. So my lab is associated more with PPARs and LXRs. And so they're both nuclear receptors. They both control energy regulation of the body. Cholesterol levels in the brain, the energy regulation.
You keep your mobile because of that.
Yeah. I didn't think of that. That might be it. Thank you.
That's the causing disturbance.
Okay. There we go. So our lab looks at PPARs and LXRs, and my postdoc is going to talk about a different receptor, or a different area. So because of the severity as well as the association of diabetes and Alzheimer's, they consider Alzheimer's almost a type 3 form of diabetes. And so the question is can we treat this with current therapeutics? So my first grad student came in and asked a question. If I improved insulin sensitivity, would I improve behavior or memory? Right? Simple question. He did that in a super diabetic animal, and he found no. The answer was no. So instead what he did was he took the drug, rosiglitazone, and he put it in a mini-osmotic pump, and he put it directly into the brain. And he showed that it did improve memory. And so it was that class of drugs which actually improved the memory and the synaptic plasticity, or the activity of the neuron. And so he published this in Neurobiology of Aging, and now he's currently working at Pfizer. So our area, we're looking at these heteronuclear receptors. What do? They come together with RXR, and they induce transcription of genes. It's a nuclear receptor, right? And so it controls genes that could regulate metabolism, but also many other, like growth factors, which help the brain and the neurons to regenerate. And that's what we're trying to do here. We're trying to understand what's happening when we come to things like inflammation, when it comes to safety, insulin therapy, everything like that in the brain, but at the same time, the safety of it and efficacy. So, rosiglitazone was removed from the market because of safety problems. And so, we decided, could we tackle that? Could we hit that receptor without causing the detrimental side effects of that therapeutic? And so we started looking and designing it. And so my lab is half medchem. And because of the medchem, we do a lot of in silico modeling. And we could look at what do these current therapeutics look like from a medchem perspective. So TZDs, or thiazolidinones, have this backbone, right? The dione, right? The sulfur product there, right? Thiazolidine dione. So that gives it the property to increase PPAR gamma. Okay, all of you guys know that, right? Insulin sensitization, pioglitazone induces that. So how does it fit in the receptor? So we did some in silico modeling, and we looked at the entrance, R1 and R2, of the PPAR receptor. And we started making molecules. We looked at every amino acid interaction link, and we created mutations to test it. What would it do, activity-wise, things like that. And we kept docking and docking. We came out with several compounds. Then we looked further as to how it fit or the AF2 ligand binding pocket. So this traditional PPAR gamma molecule will fit into entrance, then arm 1, and interact with this tyrosine 473 site. Strong interaction deep in the pocket, and it causes heart failure. It was removed from the market. So we came in and looked at it, and instead of only going into arm 1, we wanted to go into arm 2. So it wouldn't be as strong. It would be a partial agonist. And so we would avoid the tyrosine 473 site. So we kept going, and we developed this model. And it's called like a mousetrap model as to how the receptor forms together and how does it come apart. So when there's no ligand, it's very loose. And it can't form an interaction, and it causes transcriptional regulation. A full agonist would be very tight and will hold the DNA in place, cause transcriptional regulation. And we wanted something in between. So we did go further. And our compound, one of our lead compounds, AU9, avoided the tyrosine 473 site. We also wanted to improve energy regulation in the brain and decrease inflammation. So we made a PPAR delta, PPAR gamma agonist, a dual agonist. And then we did molecular dynamics. And with the dynamics, we were able to identify every amino acid and the interactions and the binding within the ligand binding domain. And we could create mutations in there to see what happens. When we make the drug, what would happen there? And so we started testing the compounds, and we do a promoter activity. So the promoter activity is like an assay where we can put it into cells and it'll light up when we hit that specific amino acid in the ligand binding domain. And so we get a dose response curve. So the guys in pharmacology, you all know this, dose response curves here. And we looked at delta-wise, we had a very strong PPAR delta agonist. And then we looked further in here, and we see that compared to GW0742, which is a really strong delta agonist, we get a similar activity. We then put our drug into a 3x TG, which is an Alzheimer's model. We found that the genes associated with energy regulation of the brain were significantly reduced, and that our drug improved that dramatically. So this kind of tells us we have a strong PPAR delta agonist, which helps in increasing energy regulation in the brain. So we went further, and we found that, hey, because we avoid the tyrosine 473, we have a weak agonist, or a partial agonist, as we see here in this dose response curve. And then when we created a mutation for the tyrosine 473, in the normal pioglitazone, we see a significant reduction, whereas ours continues, suggesting that our drug does not require 473 for activity. So we went further, and we looked for gamma activity, and we looked at, again in here, for energy regulation and other pathways, and we found we get a significant improvement in this Alzheimer's model when we compare it to an Alzheimer's model with just saline treatment. We then looked into fat cells, and so fat cells can really test us to say, hey, if we had a strong PPAR gamma agonist, we should get a lot of lipid accumulation. And our drug does not, suggesting that we're getting a partial agonist or something that's not as strong as what is compared to a full agonist, as we see here. So we've done some in silico modeling. We've tested this in Alzheimer's model. So what we wanted to do was to really test this as far as neural activity, as far as behavior, as far as further work. So one of the physiological effects, if you give a human pioglitazone, they improve their insulin sensitivity, but they gain weight, right? Adiposity, all these things happen. So the first thing we looked at in wild type and in transgenic mice, do we get an increase in weight mass? And so over 3 weeks, 4 weeks, we looked at that, and we found that the pioglitazone, the full agonist, induces weight gain, even in the transgenic animal, whereas compound 9 did not, as we see here and here. And so we looked at heart size, we see that pioglitazone does increase the heart mass, which is actually known, that it does increase the heart size, and that our compound did not induce that effect. So we're kind of happy with that. We're looking at that further and further and saying, we get an improvement in behavior. What happens? What happens? So we went further to look at pathology. And we looked at, in this animal model, which is a 3xDG, which contains 3 human mutations, 2 for amyloid beta, 1 for tau. Those are the 2 major forms of pathology associated with humans, which induces Alzheimer's disease. And so you can see here that at certain times, you get an increase in amyloid beta. At 12 months, you get a significant amount of tau pathology, as well as neurodegeneration. So what we decided to do was start giving the animal at 9 months and continue giving it till 12 months in age. So we know that they have significant pathology. And we did a novel object recognition and a Y-maze test. And we found that we get significant improvement in both of these. So this is working declarative memory. We get a behavior, so we get improvement. We then went further, and we did electrophysiology. Obviously, I didn't do this, but one of my collaborators, Dr. Vishnu Subramanian, did do this study for us. And he looked at this as far as inducing, or trying to induce synaptic plasticity. So we looked at it, and here's our control animal, or our Alzheimer's model, which can't retain the signal. And then when we gave the animal drug, you could see it significantly improved, as we see here, and then it comes back up, as we see here. So we were pleasantly surprised with this. So we then wanted to know, how is this happening? How is this actually that this drug is working in these animal models? So we looked at markers for presynapse, postsynapse, and then others in the other areas, extrasynaptic regions. And are there actual morphological change in the neuron itself? So again, we looked at all these markers. PSC95 is postsynaptic density 95, and it marks that the postsynaptic receptor region has changed. And we found that in the transgenic animal. That we get a significant reduction, as we see here. Then with our drug, we get an improvement, as we see here. Specifically, the glutaminergic receptors, GLUA1 and GLUA2, we get an improvement, as we see here and here. So we then did some transcriptome analysis. We looked at markers for neurotransmission. You see here in the transgenic animal, a significant reduction. That's the blue area. And then in the transgenic with our drug, it increased or improved almost similar to wild type. We went further and we wanted to look at what's happening. Is there neurotropic support with our drug treatment? And so we went further and we looked at the spines. So we looked at 2 regions, the cortical neurons and then the hippocampal regions. And the cortical regions, we're looking at these little spines and in the hippocampal, which are here. These represent postsynaptic regions. Do we get improvement in the postsynaptic regions? So we have software which will read our spines for us. And it'll categorize, do we have the branch chain or do we have degenerating type as we see here? And so our neurons look very similar, something like this. So we then looked at, here's a wild type with a lot of nice spines. And then the wild type with the compound 9, you get a similar pattern. And then the Alzheimer's, you see a degeneration at 12 months in the spine. And then with our drug treatment, we start seeing an improvement. So we looked at spine density, spine length, spine areas, and this is all the computer scans it gives us the data. Right? It's neural-assisted software. We're blinded to what everything is done. So what induces this increase are these neurotropics. BDNF, NGF, we see an improvement in this. And it turns out that our drug transcriptionally regulates BDNF. And we've done promoter activities, found sites specifically for PPAR delta on BDNF to regulate it. So we then looked at A beta. And here's the animal with the immunofluorescence for amyloid plaques. And then with our drug treatment, we get a reduction. And then we did the densitometric measurement. We see a reduction here. And And then we treated that, so we created a cell line called APP CHO cells. We put in the amyloid beta APP into the cells, and we were able to measure how much is secreted out. When we observed that, we get a significant reduction, as seen here. And then we observed that, hey, do we inhibit the enzyme that makes amyloid beta secretase? We found that we get a significant reduction, but we found that it's very similar to a PPAR delta agonist. And that was interesting because our drug was a dual agonist, but that it was the delta component of our drug which really is beneficial. So that's what was so important here. We looked at tau phosphorylation. So here is tau phosphorylation in the triple transgenic and then triple transgenic with our drug. This is the control. So we stripped this block, reprobed it for total tau, so that way we know that there isn't a change in tau expression levels, it's just a phosphorylated form. And we get a significant reduction as we see here. So then we look for markers for neuroinflammation. We see we did transcriptome analysis as we see here. We get a reduction in inflammatory markers. TSPO is a clinical marker, and we could see that we get a reduction as we see here. And we then did a nitrite assay. We plated microglia cells treated with an inflammatory agent like LPS. We found that we get a reduction. We then looked at cytokine expression in the brain by transcriptome analysis. The triple transgenic, of course, has high levels of the cytokine expression. Our drug brings it down, similar to wild type. That would be almost predicted because PPAR delta agonists really tremendously reduce inflammation. We then looked for more for cellular stress by transcriptome analysis. Again, it increased with the triple transgenic, and with our drug, we brought it down. Not as good as the wild type, but it's similar. So from this, we learned that we could produce a drug which is a little bit more safe, which we could get into the brain, and that we could induce changes in neuroinflammation as well as improving plasticity. So this drug is now leased out to a biotech company, and it's in phase 2 trials, non-FDA. So we're still garnering data to advance it towards an FDA application, an IND application. So what do we do from there, right? We started looking at it. We said, hey, how do we understand what's better? So we started developing compounds for LXRs. And so we did a dual agonist of LXR and PPAR. And the LXR in the brain isoform is beta. So we made an LXR beta isoform and avoided the alpha, which is most expressed in the liver. We did computational in silico modeling and screened about 1 million compounds to get this. So this class of drug, again, is transcriptional regulation, and it actually induces an efflux of harmful cholesterol. So patients which have ApoE4, this is really meant for those types of patients. 50% of all early Alzheimer's patients have the ApoE4 allele. We're trying to really target this towards these types of patients. So my 2 grad students, Ian, and now I have actually 2 more, which are working on this project right now of designing and developing molecules. So they've screened thousands of compounds, and they've synthesized compounds from all different directions and developed a whole bunch of different compounds, and found that we could develop this one compound, 403, and we could induce that to have specific activity for beta. We then produced compounds, we screened them, we found that the LXR beta, 45 nanomolar, for alpha, is 450 nanomolar. So it's really good separation, tenfold. Current compounds for LXR class induces neutropenia, so they can't use it clinically, and it induces liver toxicity. So we gave it to the animals, and we found we get no lipid accumulation. That we don't get an increase in cholesterol levels in circulation when compared to the other compounds in the class. We looked at liver cells. 2 of the compounds do induce lipid accumulation, but these are neutral lipids. Our compound did not do as much, so we were happy with that. We then looked at beta activity, again, was really good, alpha was reduced. So we looked at neutropenia. We found that the neutropenia doesn't occur in current erythroid cells, but then in bone matrix cells, we can see that GW, which is a traditional compound, induces toxicity, and ours doesn't. So we could avoid the toxicity associated with traditional LXR compounds. And that was a real game changer because now we could target patients which have ApoE4. With that said, we then gave this to triple transgenic. We saw, here's the amyloid beta plaques. We got a reduction in the plaques. We then scanned them, reduction. In the APP CHO cells, we see a reduction. We get a decrease in tau a little bit. So we were happy with these results. Dr. Vishnu, again, did some electrophysiology for us. We get an improvement, as we see here. It wasn't as good as our compound 9, but we still get an improvement. So we observed that we get an improvement in preventing the liver toxicity with the traditional LXR compounds. And we went further and we looked at gene targets within the brain, and we found that we get an increase in the transporters for cholesterol, so we're effluxing out the bad cholesterol. And that's what we saw when we did the behavior, that we get a significant improvement in behavioral patterns in these animals. And that was it. So I gotta thank my people. We presented this data at the Alzheimer's Association International Conference in 24. A lot of fun going to that meeting in Philadelphia. And then of course, we gotta thank our people, all the guys in the lab who have worked so hard. I have my daughter now working in the lab here. So it's free labor. You know, she works in the lab. It's good to have a new dishwasher in the lab always. And then I have to really thank our department and the support from our college of Pearson School of Pharmacy, our College of Pharmacy, and our department for drug discovery and development. I can take questions.
Yes, thank you, sir. What an enriching and thought-provoking session. On behalf of organizing committee, I extend our sincere gratitude to Dr. Rajesh Amin for his outstanding presentation on design and development of novel LXR agonist for Alzheimer's disease, along with I think PPR gamma agonist as well. Dr. Amin, your talk provided insights into the complexity and opportunities, like with the help of PPR gamma receptor agonist, you developed the LXR. To be precise, selectively the drug is targeting to the LXR beta. This is a kind of opportunity we can say for developing the therapeutics for the neurodegenerative disease. The innovative compound that is AU9, specifically on the PPR gamma receptor, and AU403, which is on LXR beta receptor. You introduced with its ability to balance efficacy and safety. It is a testament to your expertise and vision. Your work not only addressing the significant medical challenges but also inspiring the next generation of researchers to push the boundaries in pharmaceutical science. Thank you for sharing your knowledge, experience and passion with us. It has been a privilege to host you and to learn from your valuable research. Now the floor is open for question and answer. Okay.
Yeah, you told about nuclear receptor. There is a FXR, farneside X receptor, RXR. They are mainly involved in the synthesis of steroid, that is cholesterol. So, how we can direct with the help of these receptor to the brain?
Yeah. So the patient class that this compounds are meant for are for people who can't transport cholesterol properly, specifically LDL. And traditionally, people have used this class for atherosclerosis. And the reason why is that in the heart, you want to get rid of the cholesterol, so the macrophages in there don't become inflammatory home cells. We made these to cross the blood-brain barrier as well as affect beta, not alpha. And so in the heart and those areas, it's more LXR alpha. So we wanted to hit just the beta, the efflux cells, bad forms of cholesterol, which then helps microglia and other cells remove some of the amyloid beta and tau products. So they've shown that in literature, when you overexpress LXR and things like that, you could clear away some of these harmful products in the brain.
Yes, any more question? Okay, sir, thank you.
That was an incredible session. Thank you so much, sir. I request you to kindly mark your esteemed presence by signing on the board. I request Dr. Uttam More to felicitate Dr. Rajesh Amin with a memento as a token of love and gratitude. And I request Dr. Dharavashi to felicitate Dr. Uttam More with a memento as a token of gratitude and love for chairing the session.
मिनट दिख रहा है, हो जाएगा लेक्चर।
लास्ट लेक्चर है वो।
Moving on to our final session, which is structure-based computer-aided drug design to identify potential lead molecules for asparginal endopeptidase inhibitors by Dr. Meenakshi Singh, postdoctoral fellow, Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University, USA. We have Dr. Samresh Pal Roy, Professor, Department of Pharmacology, School of Pharmacy, BP Savani University, to chair the session.
Hello. Thank you, Setaman. So, ladies and gentlemen, it's a privilege to introduce our distinguished speaker, or we can say the last speaker of today's 7th International Conference, Dr. Meenakshi Singh, a postdoctoral fellow in the Department of Drug Discovery and Development at Auburn University, Alabama, USA. Dr. Singh is a highly accomplished researcher whose expertise in natural products chemistry and computational chemistry has significantly advanced the field of drug discovery. Dr. Singh completed her PhD at NIPER, Kolkata in 2021, specializing in natural products. Her academic excellence is evident in her achievement securing the top rank in GPAT and NIPER JEE for both her master's and PhD program. She has also been honored with the DST Inspire Fellowship in 2017, underscoring her remarkable dedication and contribution to research. Currently, her work focuses on designing and developing small molecules targeting asparginal endopeptidase, a promising therapeutical approach for neurodegenerative disease such as Alzheimer's disease and frontal temporal dementia. Leveraging structure-based computational aided drug design, Dr. Singh has identified potential lead compound that could revolutionize the treatment for this challenging condition. So today, Dr. Singh will share her research in a talk titled Structure-Based Computer-Aided Drug Design to Identify Potential Lead Molecule for Asparginal Endopeptidase Inhibitors. Her work not only shed light on the innovative therapeutic interventions but also lays the groundwork for addressing some of the most pressing healthcare challenges of our time. So let us give a warm welcome to Dr. Meenakshi Singh as she shares her innovative research with us. Over to you, ma'am.
कुछ नहीं आ रहा है भाई 37 कॉपी करके ये डाल दो पार्ट 1 पार्ट 2 लास्ट वाला
Now it's good. Not yet. Okay. I'll use now. Oh. Things happens. So thank you for being here, and I know it's very hard to have to post lunch, be here, be attentive, and listen to talk. It's a long day, so let's start. I will finish in very short and very crisp this presentation and what I'm doing from past year under the Dr. Amin presence. So my title is to design inhibitor for neurodegeneration, targeting the delta secretaries in Alzheimer's disease and frontal temporal dementia. So the first thing comes in mind, what is Alzheimer and what is frontal temporal dementia. So you see there's about 69 million people are affecting from this Alzheimer, and if you see the growth, there's a highest number in the 75 to 84 years age people have suffering, followed by this 85 plus and 65 to 74. You cannot say that this is an old age disease, okay, because it can occur at any point of your age. And if you see that males are more prone for people. Is a loss of your behavior, your social, and your way how you behave with the circumstances. So let's start understand what is the dementia. Is the umbrella of the disease is come with all this Alzheimer, vascular dementia, frontal dementia, and Lewy body, which comprise and give you the list of this. And if you see, we are working on first Alzheimer's disease, frontal temporal dementia, Lewy body dementia, vascular cognitive dementia. And if you mix Alzheimer and frontal temporal, you can say that it has a mixed dementia. That means you will get the both behavioral as plus as cognitive decline, social impairment, everything. So let's start type of dementia in Alzheimer, and this is frontal temporal dementia. So this part is a frontal temporal, which regulates your planning, organizations, personality development, and social gathering. This is temporal lobe, which is basically for your memory and language, and this parietal lobe is for the thinking, that we have to get the good marks. So because of this, it's recognition, sequence, and sensation. And this is the optical lobe, which is meant for your visualization. So what happened during your Alzheimer's disease and dementia? You always think that we have neuron to work with our brain, but not. Not that only the neuron is responsible. We have a certain different component which are regulate together to make our thinking better and all those things. So we have the oligodendrocytes, astrocytes, and microglia. Basically, these are the means you can say the oligodendrocytes is a helping in a myelin sheath. What is a myelin sheath? Myelin sheath is conducting, like you can say that's a neuron and there's a myelin sheath, and which is helping in the conduction. Conduction is very important when you have the transmission, right? And then your astrocyte, which maintain your homeostasis, which maintain your ion balance in the brain. If the ion balance is lost, you lost everything, because it's very important. You always heard about the homeostasis of the body. So So if the hemostasis is unbalanced, you will lose everything. And the microglia is a phagocytosis which clear everything which is not unwanted in the brain, and the neuron. They all come together and function, making brain work. So here I am giving you a brief things which are responsible for the Alzheimer's disease, like amyloid beta, as all Dr. Amin has talked about, and the tau and TDP-43. They all make this condition worse. And if you consider this tau and TDP-43, this gives you frontal temporal dementia. If you consider this amyloid beta and tau, it will give you Alzheimer's disease. So how to cure this? Is there any drug in the market? Is there anything which can cure all these symptoms early? Yes, there are. There's some, but not many. But they are not US FDA approved for cleaving the property which I just talked about, like tau, amyloid beta, and TDP-43. And there's some monoclonal antibody, and you all know that these are not good. Protein peptides are not good, which are not compatible with our body. So, is this the really best we can do? I think we can do much more than that. And how we can identify the potential drug candidate. And I feel like before identify the potential drug candidate, we have to identify which is the best target, how we can target best so that we can cure the disease. So, in this, in my postdoctoral training, I am working on this delta secretase. It is basically we can call it, you can call it as legumine, you can call it delta secretase, you can call it asparagine endopeptidase, and it is a lysosomal cysteine protease which cleave the protein which I just talked about, like all these protein. It cleaves tau, it cleaves TDP-43, it cleaves amyloid beta, and cleaving all these things make you fibril, plaque, and the tau tangles and this aggresomes. So what happened? So this is an inactive state. At present, this is present in your body, but it is inactive, right? When it's active, when you aged, or when you feel some stress is coming, when you're very long time you are very ill, so our brain pH decreases. So when the brain pH decreases, this lid is come off and then opening to the active site. What is this active site? Active site means this side is going and bind to the various protein and makes them cleaving and makes them clot, and that is not good for us. So why it is important? So it is important because when the level of this enzyme increases, it cleaves all set of all these proteins, which is considered as SET, tau, ABP, CHULS, 7/9, TDP-43, and alpha-synuclein. And we are targeting particular 3 of them with this delta secretase, and we are focusing on AD and FTD. So how does this cleave? It's cleaved by amyloid beta, this is TDP-43 and tau. So these are the cleaving site by the different enzyme. You always read about that secretase, beta secretase, gamma secretase, alpha secretase. So there is another one which is delta secretase. And if you see this N-to-C terminal, it is cleaving right in the first term, and then followed by the second also by the delta secretase. If you have a drug which can cure or inhibit these things, you don't need to go to the beta secretase or gamma secretase because they are coming later, right? So we need a drug which can target right here. And then if you go for the TDP-43, it's cleaving the glycine-rich region, which is responsible majority of the FTD and AD. And right here it is cleaving from this point. And in tau, it's cleaving from 255 and 368 region. So that is why this delta secretase is so important to target about, because it's cleaving the first site of the amino acid in the domain. Now it is a complete picture how it is going. So this is a neuron that I just talked about. This is the myelin sheath, oligodendrocytes make it, and there's some microglia comes and take clearance of the protein which are not needed. Here's a TDP-43 which is located in the nuclear region. This is the APP which is the amyloid processing protein which makes your amyloid fibril, and this is tau and which makes your tau aggregates. So when disease happens, so this TDP-43 misnucleates and goes to the plasma, and this which activates the microglia, and this happens, all this degradation happen, and your neuron is damaged, your conduction is damaged, your transmission is damaged, and your brain will not recall whatever you have done just 10 minutes ago. So this is a disease state brain and this is a healthy brain. You can see the difference between the disease and the health, right? So, and that one FTD is typically faster than the Alzheimer's disease. So what are the approaches? Like, I don't know how many people you know about this structure-based drug design in silico properties. So we followed in our lab as a drug discovery protocol. So we followed this as structure-based drug design, where we screen billions of chemical space, chemicals, functionalizations through the various software which we have used is Schrodinger. We select a target and we screen large databases, and we put various selection like propensity rule and everything, and we find doc and then we see the interactions. And this is some literature which impact on this data secretaries is very important. There's a paper which very few papers are there for the data secretaries, and how this asparagine cleaves TDP40 in the brain. So the research goal and significance of our study is to address the unmet therapeutic needs, and then how this we can develop a very good, we can see the candidate which can integrate the protein aggregation and misfolding, and how this structure-based drug design we can use to get a good candidate. So this is the working model of our work. So this is asparagine allopeptidase domain. If you see there's a 3, I put the star, which is, I talk about the active sites. So this is asparagine, this is histidine and cysteine. These are the 3 important targets which goes to the various protein and then makes degradation. So how we have to cure it? So we have to just block it with some of the drug which can help to make its data secretaries. So we focused on this, and we screened CNS focused online database, more than the 20 millions of compound, and we did all these things at high throughput screening. We identified top 20 doctrine, and then we have done this ADME properties, and we finally published this in a good journal. And this is a kind of interaction study, which if you see that I just talk about the asparagine 42 and histidine 148 and cysteine 189. If you see, the most of the molecules are going to interact with this particular thing. So we screen it and we finalize this top 5 compound for us. And if you see this hydrogen bonding and the salt bridges and the pi-pi interaction with the histidine and the water bridges and the pi-pi cation interactions. And this is a compound, 11 compound, which is recently done by the Emory School, but we could not find this a big good compound because it doesn't interact with any active amino acid residue. So we run this multi-dynamic simulation. I don't know how much you know about the simulation. It's a real-time simulation because in our body we have always a dynamic things, protein and water. So we done this compound 11 and we found this is not good because if you see the light in RMSD is quite high, like 65, which is not permissible, and if you see this protein RMSD also not matching, it's not stabilizing the complex. So it is not a good molecule for us to go ahead. So we ran for our identified molecules, and you see the drastic change from here to here. Let's see how this complex is quite stable with the compound. For the API 1, 2 and 3, we all and getting a good interaction as well. So then we run the PCA and we have done this energy, field lattice energy, and in this we have to see that how conformational changes happen with our molecule and the asparagine allopeptidase, and we see that throughout up to some time our compound and the protein is get stabilized. But in case of compound 11, you didn't find that it is stabilizing throughout, as we see on this multi-dynamic also. Then we have done the comparative study where we have to identify the IC50, and we got that our compound, the highest is this 0.351 micromolar, and the compound 11 is 0.7, which is double of it. And we ran the Big-Higgs-Manton equation also, and we found the Ki of the compound is 1.0 0.102 macromolar, and then we have done some this estrogen and endoperoxidase activity, and we found that with a compound we are decreasing the delta secretase activity, and we done the ELISA also. ELISA is basically we have done for the amyloid beta, and because this is a toxic fragment for the amyloid beta, and we found that compound C, our compound is SC is decreasing by the half of the fold as the control and other compound and the compound level. So this is our future direction. We have to test this compound and compatible with the ATME, because this is a very, in a very nutshell, it's just started. So we have to go in a deep study, and we have to see that how it is behaving with the mass spec, and we have to see that ATME profile. We have to make the crystallization of this, and for this toxic studies and the PDP. This is my lab, and I have to acknowledge Dr. Rajesh Amin and the Harrison College of Pharmacy. And this is my lab mates. Ian is graduated, Zar, and Samitra, Nick, all of them. This is our building where we work day and night. Thank you. Very short.
Yeah. Thank you, ma'am, for your crisp presentation. I really appreciate your insightful session on this topic. As you rightly mentioned that how exactly we are targeting the delta secretases to inhibitors to target the tau protein, APP protein and TDP43. And also you have rightly identified the structure-based drug design for getting a good molecule to target this disease. So now the session is open for question answer. If anybody want to ask some questions, you can please shoot your question. Yeah, anyone would like to ask some questions? Because ma'am has rightly discussed about the, I think, basic part of dementia and Alzheimer's disease. All of you have studied in your B Pharm as well as M Pharm. What is NDD, how Alzheimer's disease developed, what is dementia and all. So and later on she tried to develop new molecule to target this delta secretase inhibitors. It was really very inspiring session. So any questions from participants? No one? Okay, so fine. So thank you ma'am for your kind session. So on behalf of the organizing committee and everyone present, I extend our heartfelt gratitude and best wishes for your continued success in this critical area of research. So let us give a big round of applause for a remarkable contribution to this conference. Thank you everyone for your active participation. I look forward to seeing you in the upcoming sessions. Thank you ma'am.
I request to ma'am, kindly make your mark on the board. And I invite Dr. Samresh Palroy sir to hand over the memento to ma'am as a token of gratitude and love. And I request Dr. Ms. Achla Vyas to felicitate Dr. Samresh Palroy sir with a memento as a token of love for chairing the session. At the end of the session, we come to the realization that knowledge indeed has no end. Today, we witnessed an information symphony that will undoubtedly value add to each of our professional and personal life. A big thank you to our esteemed speakers, participants and organizing team behind the scenes. Thank you. And one cannot pour from an empty cup, so let's refill ours with high tea. Before we close, I invite you all to be a part of the cultural and award night commencing at 6 p.m. in the lawn. So do soak up some culture and cuisine of Gujarat. So wishing you a great evening. Bye then. Myself, Dr. Shweta Swaminath, signing off. Thank you. all, kindly fill the feedback link which has been shared in the WhatsApp group. It's a request that you all of need to fill the feedback link which has been already circulated.
Ma'am, can I have your number? Okay. I'm going to come to meet you.
Sure.
Morning I'm searching. Where is she? Where is she?
Oh, from morning
Sweta.
you were here. Okay.
Morning is after 1 p.m.